We have a presentation coming up next from Bobby, is it Bobby from the Clock Foundation anyway?
It's Bobby Brooke.
Bobby Brooke, thank you. I had a mental lapse. Hopefully there'll be pills for that soon. Now I wanted to explain though that they are an outlier in that the Clock Foundation is not a presenting company seeking investors or partners or customers or what have you. But we started this, I don't know if you call it a tradition last year, where they were doing such an interesting and important work. This is to do with epigenetic clocks and trying to validate the range of them and looking for more data in support of them, that we put them into the showcase, and we're repeating that practice this year. So Bobby, if you're there, would you mind showing yourself and sharing your slides?
All right, here we go. So, my name is Bobby Brooke. I'm a co-founder of the Clock Foundation, or the Epigenetic Clock Development Foundation, with Steve Horvath from UCLA. I got to know kind of him and his work through a clinical trial that I did with Greg Fay, who's an advisor to the Clock Foundation, and and that work was focused on thymus regeneration or immune system regeneration in healthy older people. We did a study at Stanford that we found was able to improve different immune system biomarkers and also reverse all established epigenetic aging clocks to a degree. And we're now doing a larger study called the TRIM-X trial. It's about a 100-person study, and we're enrolling people across the US right now. But with the Clock Foundation, we also have a nice group of epidemiologists and biostatisticians, kind of data science heavy group, as well as people that are focused on like cell biology, preclinical studies, and have experience with clinical trials. And really kind of the goal of the Clock Foundation was to spin out technology from the Horvath lab and make it available for different uses, for drug development, for mechanistic studies, for kind of health monitoring in canines and humans. And we've had a pretty busy last year, and we did over we have over 75 projects that we've done with different groups. And this is kind of a mix of preclinical and clinical studies, as well as studies that are just looking at the mechanisms of aging. So kind of basic research questions that we're trying to address, mainly through DNA methylation and epigenetic clock studies. And this figure, this figure's from a paper by Steve Horvath and his close collaborator Ken Raj, and and it just kind of emphasizes that one aspect of what we're doing are just kind of supporting studies on kind of the fundamental biology of aging, and trying to address some of these questions about like what actually makes up the epigenetic clock. What does drive the epigenetic clock, and how does that kind of play out or manifest in aging, or how does that affect organismal aging? And so, like, you may be familiar with epigenetic clocks and know that like induced pluripotent stem cells and embryonic stem cells have an epigenetic age of about 0. And but kind of primary cells, of course, do kind of age in vitro, and so there's kind of different ways you can kind of look at epigenetic aging. I mean, you can look at it in vitro, you can look at it in tissues and organs, and also, of course, at the organismal level, which a lot of people are focused on, just taking kind of blood samples or saliva or cheek swabs. And then another aspect is that we're focused on using these biomarkers for treatment development. And there are many companies. I saw Loyal was presenting just before this, who's doing exciting work with canine longevity. And there are a variety of different groups developing longevity drugs and treatments, and everybody wants kind of reliable biomarkers. They want some feedback on what's working or what's not, and epigenetic clocks appear to do a decent job of that, and so they can be used in vitro, so they can be used in cells in a dish, and for kind of mechanism of action studies. They can also now be used in a variety of different preclinical studies. I'll get into this a little bit more later, but they can be used in rodents, they can be used in dogs and cats, and also a lot of other mammalian species. And I think most people are kind of familiar with how they can be used in human clinical trials and in human health monitoring. There are different clocks that predict chronological age, and there's also newer versions that are more calibrated or geared towards predicting future health outcomes, predicting future morbidity and mortality. And so if you're a drug company, or you're a nutraceutical company, or a physician or a clinic developing or optimizing treatment regimens, there are these tools available now, at every stage, in vitro, in vivo, and then on to clinical trials, where you can test these interventions and really, kind of get a pretty good sense, kind of, about how effective it is, how reliable that treatment is, and it's kind of our hope that by making these tools available, it'll lead to a lot more, kind of a higher success rate in translation, and just kind of more reliable interventions becoming available. And one epigenetic clock that I want to highlight is GrimAge. So this is a predictor of, is kind of calibrated as a predictor of mortality in large population studies. And it really kind of sets a high bar for epigenetic clock, or for aging biomarkers, kind of generally. It's and I mean, it is published. That's something that of course is very important. When you're it's also been replicated by independent labs. It's been shown to have the same links to morbidity and mortality, even in large diverse populations. So it's not something that's kind of kind of overfit to a small population or to a certain sample type. And then also there's been kind of this reliable link to different health outcomes, to different disease outcomes. So there's like the major age-related diseases like time to heart disease, cancer, and there's also a link to improved cognitive health, or one interesting study showed that GrimAge acceleration was linked to PTSD in a large VA study. It was done at Walter Reed, and there are also kind of more recent studies that came out kind of over the last year or two that show a link to other kind of clinically meaningful functional outcomes, things like walking speed. And so like GrimAge is available for clinical trials, and there's also now been work done in the Horvath lab over the last several years to develop epigenetic aging clocks for all mammals. And he developed a mammalian array. So it can be used, it's now been used in over 200 different mammalian species. And there have been some pretty remarkable results. And many people are familiar with the Horvath pan tissue clock. That was a 2013 publication that discovered a clock that predicts age across all human cell types and tissues. And so this is kind of going a step beyond that, and there is now a universal pan tissue mammalian clock that predicts age across all mammals and across all cell types and tissues. And so there's also now predictors of maximum lifespan, based on this. And it's starting to enable different applications, like use in rodents and preclinical studies, also companion pet studies. And yeah, I guess I can highlight some of the research that, there's a lot of different collaborators, as you can imagine, with the study, with kind of work like this. But like, there's people that are now profiling cattle and horses, and then a lot of the species that are exciting, that have exceptional longevity, like naked mole rats, bowhead whales, up there in the upper right, you can see. One area that I guess Loyal just talked about, and others are very interested in, is companion pet longevity, companion pet health monitoring. And so there are now highly accurate epigenetic clocks, so highly accurate in the sense they can predict chronological age very well, with both dogs and cats. This is their publications that came out this year. The canine is just a preprint at this stage, but they are useful for epigenetic age monitoring and companion pets, and they can also be used across different sample types. So a lot of testing has been done in blood, but there's also preliminary data showing that like a more convenient sample collection methods could be used. So like an at-home collection method could be used for both cats and dogs. And this slide just kind of gets at this question of like direct to consumer applications of these clocks, like many, and so like, what's the distinction, like with a 23andMe type company, they're offering genetic testing. There's a lot of people who see DNA methylation and epigenetic clocks as kind of this next wave, of where people can, it's something where people can get tested year after year potentially. And it's also something that, unlike your genes, it's kind of more easily modified, by diet, by exercise, by kind of different longevity treatments, or and so there are a variety of groups that are kind of focused on this, again, for human health, and also kind of canine companion pet testing. There are also very are pretty reliable, I'd say, and scalable methods, or kind of tools available to do this type of testing. There's array technology from Illumina, and kind of systems and liquid handlers that can be obtained pretty readily, where people can implement them. And in that said, I also, when you're talking about doing consumer testing, it's like an individual test. These are research use only biomarkers today, so they are kind of investigational use tests, and still today it's pretty expensive. There's some work that's underway right now at the Clock Foundation and with others to really help make the testing even more accessible, to sort of really kind of drive down the costs and to make it more reliable. And but I feel like like we're pretty close to being at that point. So I think over the next 12 to 18 months, you're going to find that the cost of this testing will go down, maybe quite a bit. And and I think it's just going to get kind of more and more informative for individuals that are doing the testing. And I think even today, it's already very helpful for decently sized clinical trials, 10 or 20 people. You can do pretty good statistical power there. And one role of the Clock Foundation is just to make these tools available, and to kind of raise awareness about them. We want to really enable teams wherever they are to teams that are focused on aging research and developing aging interventions to be able to use them. And so we've done a lot of work over the last year setting up or kind of helping to coordinate testing infrastructure and clinical trial infrastructure in the US and then also in the UK and Europe. And so we're also taking steps to where it's pretty scalable, where groups can pretty readily not only do a trial with 10 people or 100 people, but go up to the thousands or tens of thousands. And we would be looking at not only epigenetic clocks, but there are a variety of other measures that can be done pretty convenient. Also, like safety monitoring is a big component of that as well. In this slide, I'm just highlighting a paper that I came across and that I liked, and I kind of compare to maybe the challenge of developing an aging treatment, something that actually kind of counteracts the aging process and can do it safely. And it's this paper about the Wright brothers and what were kind of some of the success factors. But it also kind of, I think, speaks to how we're trying to make tools available that aren't just in the hands of big companies, but also small teams. And there's a lot of great teams out there, that are very focused on aging research, and we're living kind of in a time where we have these great biomarkers and analytical tools that are available. And we really kind of overall, we just want to put these tools in the hands of teams that are focused on developing exciting aging treatments and empower them. A longer term goal, I think this is probably a 3 to 5 year goal, and maybe one for the whole industry is to kind of address this question of why is big pharma not even more focused on developing longevity drugs or developing drugs that can be used in a preventative setting. And of course, one of the answers is there just is no regulatory path for it, right? There is no clear regulatory guidance about how to develop a longevity drug in a preventative setting. And so I really appreciate kind of all the work that Thomas has done kind of in this area. Just kind of he's had a nice biomarker panel a couple of days ago, and then people from he's engaging with people from FDA and getting them kind of talking about it, and that's a tremendous step. And and one approach that we're taking at Clock Foundation and through work with treatment developers is to kind of long-term target use of aging biomarkers as a surrogate trial endpoint. And it's a special type of biomarker that can be used to gain kind of conditional approval of a drug, but it's still going to be contingent on kind of long-term safety, long-term outcomes data. But that's really a process that we're kind of longer term and we're focused on. And I have this question about here: will we see an FDA division for aging and preventative medicine? I don't know why not. I think it's kind of it's something where 10 years ago the only kind of treatments for preventative medicine were kind of diet and exercise. But now when you have metformin and you have rapamycin, you have all these these other kind of putative longevity drugs, it really does fall under the purview of FDA, and they're going to need to address it. and hopefully before too long, I mean, there will be, a whole team of people there, maybe an entire division that's focused on it. And finally, I'll just wrap up by saying that, if you're interested in using any of these tools, if you're interested in enrolling in trials or coordinating trials, please reach out. We're happy to work with a variety of different groups, and I'll just leave it at that and open it up for any questions.
Thanks, Bobby. Thanks for that presentation. I know we only have a minute or so. Just a couple of quick things. If you had a guess, what percentage odds would you put that the FDA would accept biomarkers as surrogate trial endpoints in the next 3 years or 5 years? What percent? How likely is it?
I mean, I think they're already using different biomarkers like as surrogates. I mean, they're going to require long-term safety data. Like there are things like kidney function that people can look at. It's.
I think Dennis's question is clocks and aging biomarkers, because
Yeah.
you're right, they do have surrogate markers. Like, is it Pretigo or somebody was talking about interocular pressure for glaucoma? That's a surrogate marker, but aging clocks.
Yeah, I'm sorry, that's what I meant. Yeah.
Like a percentage. I don't. Yeah. I mean, it's.
High likelihood, medium likelihood, low likelihood.
I mean, I think medium likelihood. I think it takes engagement with them, right? And that's kind of all we can do. And I mean, it also, it's. I feel like the biomarkers are ready, but the treatments, need to kind of link up to it, right? I mean, you need treatments that can modify these biomarkers, and that that actually that the groups kind of have ready to be used. It's like kind of a companion test, right? So but I think they're very open to it. I mean, all these conversations we had, I mean, that but they're also very data driven, and they require clinical data kind of so it's I mean, it's yeah, but I feel like the path is available. So.
Yeah, and just one quick last thing. One of the things that's happening with disease foundations these days is that they're taking equity in companies that they partner up with or join up with. Does the Clock Foundation do that, or have you ever thought about that?
Interesting question.
I mean, I've thought about it, but it's it's. I mean, it kind of, yeah, it's, I've thought about it, but, we're not, it's not like us, kind of a part of our strategy, so to speak. I mean, we're, we're trying to provide kind of relatively low cost kind of tools for different groups. And yeah, we're more focused on just, helping them develop exciting aging drugs and treatments, above all else.
Got it. Great. Thank you. Thanks for the presentation.
Thanks, Bobby. Thanks, Dennis. I think we should move on to next company.